Okay, hello everyone, and welcome to the second online lecture. Our today's speaker is Dr. Boštjan Vihar. He is a postdoc at the Faculty of Medicine in Maribor and Biomedical Researcher and Bioprinting Developer at the IRNAS Institute. And today's topics is bioengineering or opportunities for customized technology. Boštjan, welcome, and the floor is yours.
Yeah, thank you very much for inviting me and for the introduction. It's nice to be here. I hope next time we can meet in person. That would be much more fun. Okay, so I will share my screen now. If I can, okay, I have to do it like this. Opa. Can you see the presentation? The main window? Yes. Or the side window? Okay.
It's perfect.
All right, great. Yeah, so today I would like to talk about biofabrication mostly and custom technology that we do also for tissue engineering at IRNAS and the Faculty of Medicine here in Maribor. So to give you a brief introduction to who we are and what we do. So IRNAS is an innovation lab for custom technologies. So what that means is that we try to do complete development of prototypes. So really from concept idea to manufacturing a small series. So this includes mechanical engineering, electronics engineering, software engineering, manufacturing, of course, and testing and implementation. And there's really three main verticals in which we work. And one of these is also research equipment for biomedical research. So what you see here in the images are, for example, our 3D bioprinter Vitro, some custom perfusion systems that I will be talking a bit more later on, and also some medical devices, pulse oximeter, ECG. And then the other institution I work at is the Institute of Biomedical Sciences at the Faculty of Medicine. Here we also, I think you could say, work at the three verticals. So one is establishment of primary cell cultures and their characterization. So we work together with University Medical Center in Maribor, where we get also tissue samples, and from these we try to make to isolate cells, make stable cultures, and then we characterize them to see which kind of cells they are and use them then for further research. The second aspect of our work is focused on the materials. So usually we work with natural polymers such as carbohydrates or proteins. So examples of that would be alginate, cellulose, collagen, fibrin, and their derivatives, from which we also try to make hydrogels usually or coatings for other applications. And then we bring these two fields kind of together and use them for making tissue engineering scaffolds, building in vitro models of tissues and diseases, but also drug delivery systems and other interesting things. Okay, so to get more into the main topic of today's presentation, so tissue engineering is kind of a very popular topic today in research. There's really a lot of people that are working on that. And if we think about why this is really so interesting now, apart from that we now also have many more tools available to do this, is if you think about the many issues that we have as a society in general. For example, let's say organ transplants. If you look only at the numbers in Europe, for example, there's roughly 30,000, I think, annual transplantations of kidneys only. and 70,000 people that get on waiting lists. So the demand is much higher than there's actually possible supply. And even if you get an organ, then there's still a high chance that you will reject the organ without immunosuppressive therapy, which has its own drawbacks. So if you would be able to make tissues, even from, let's say, patients' own cells and tissues, that would drastically improve this kind of issue. But this is still really many years in the future, still from now. But even today, there's many applications where tissue engineering can be used for example, making simple models of tissues, or let's say organ-on-chip solutions where you can test new drugs or investigate how diseases work. This is still very useful, especially if it's more comparable to how the actual body works compared to, let's say, standard cell cultures or even animal models if you're using human cells. So a nice example of that is what you see here in the top right corner. I don't know if you're familiar with this work. So this is an example of a tissue engineered trachea. So basically the tube that goes to the lungs. And this is a model, for example, that was kind of used to test asthma and what happens if you add, let's say, antihistamines and stuff like that. So very, very interesting stuff. And of course, what you are probably most interested in today, also cellular agriculture is also one of the main fields that will benefit from tissue engineering. So yeah, the way agriculture and especially livestock farming works today is, we know, not sustainable and comes with a lot of problems, ethical, so yeah, healthcare related and ecological. So there's a lot of reasons to actually go into tissue engineering and do research in this area. Okay, so why do we need to go into the third dimension from standard cell culture? So the standard model of culturing human or other animal cells is really not new. This has been here for decades now. You're probably familiar with that. So how it works is you have a tissue sample and you break it down into individual cells into a suspension, and then you put them in a plastic container like this. And then the cells attach to the bottom and they spread out, and you can grow them, they multiply there, and then you can do experiments with them. So the main problems of in vitro models like this, even though this is still probably the gold standard today, is that the cells, when they're cultured like this, don't really behave the same way than they would in native tissues. And if we look closely at how these cells grow in here, we can also understand why, I think. So in native tissues, cells are, they are supported by the extracellular matrix, ECM for short, usually. And they have also connections to other neighboring cells from all directions. So chemicals can travel in all directions. They have structural support from all directions, usually, except maybe for epithelia. And this is not what happens here. So here you have cells that really attach to a plastic bottom. There's no exchange of any kind through the basal side. All the exchange of chemicals goes through the apical side with the medium, which is usually also static. And the only interaction between cells is here on the lateral sides. So direct contact, of course, you have, if, I don't know, cell 1 produces something here, it will slowly diffuse there as well, but this is not really representative of how it works in native tissues. So if we go to the third dimension, we kind of solved this problem. So we get cells to be surrounded by their neighboring cells, and you have much more similar conditions and more relevant results also for research. So there's two, let's say, main approaches to 3D cell culture. The one is this one on the left, so scaffold free. So how this works is really you have the cells in suspension, and then you prevent them from attaching to the bottom, or only allowing them to attach to each other, and then they form these kind of spheroid structures like you see here. And the other approach is scaffold based. So what you do there is you take, let's say, an ECM replacement, and then you mix it with the cells into a scaffold and build a scaffold out of them. So you give structural support, structural support to the cells, but still allow them some mobility and chemical exchange and interaction with neighboring cells. So yeah, this scaffold-based approach is also something that I'm focusing more on in my work, and so I want to go more into that direction. There's many approaches into how to make scaffolds. So some of them include non-automated techniques, like you see here. So these could include freeze drying or phase separation, knitting. Very popular is also the use of decellularized organs or tissues. So how this works is you take some piece of tissue or an organ, and you rinse it with salts and detergents to remove all the cells until you're left only with the ECM, basically, and then you can repopulate this with new cells, which is really nice because you have, you know, all the microstructures and the right type of ECM already there. But the problem is that you kind of still need a donor organ or tissue to use it for that kind of purpose. And then we have the automated scaffolding approaches, which includes 3D printing, but also other stuff like machining or laser ablation. But today, so at least from what I can see in literature, 3D printing or additive manufacturing is the most popular one used today. And I would like to maybe clarify some of the definitions, because in the last 2 decades we've had so much research been done and so many new techniques tested that I think there's kind of an inconsistency maybe in terms of how the terms are used. So just to give you a brief overview, so you're probably familiar with classical 3D printing itself, right? So you have a model of a 3D object, you slice it into individual layers, and then you basically 2D print them like you would layer by layer until you stack up the whole structure. And with transferring it to let's say to biomedical engineering or bioengineering, this still works the same, but we have new materials and also implement cells. And we have some new words that have come up with this. So biofabrication, or maybe I can refer you to this paper here by Grohl et al. This is really this has a really good set of clear definitions set up. So if we just go briefly over them, so biofabrication would be like any use of automated approaches to make biofunctional structures. Bioprinting would be like the cross section of additive manufacturing and biofabrication. So the 3D printing techniques for making biofunctional structures. Bioassembly, for example, would be if you have preformed biological structures and then you put them together and let them form after that. Bioinks would be materials that have cells implemented inside of them. Biomaterial inks are just materials that can be used for scaffolds but don't necessarily contain cells themselves. So this is just a brief overview to go to the topic of the bioprinting itself. So I would like to talk about three main categories of bioprinting today. The most I think easily understandable is extrusion-based bioprinting, which is very similar to let's say FDM 3D printing, as you would know from fab labs or maker spaces. So you have material in a cartridge and then you extrude it through a nozzle, and if the nozzle is moving over a surface, then you deposit material. And this is a very interesting technique. It's probably the most widely used of them all. It's relatively cheap. It is compatible with a really broad range of materials. So you can use hydrogels, silicones, other types of materials. But with new approaches, you can also make very interesting structures. So for example, an interesting technique is the so-called FRESH technique, which stands for free-form reversed embedding of suspended hydrogel. So basically what happens here is you deposit material into a support bath, and even if the material itself wouldn't, hold its weight to be stable, like you see it here in the video, you have the support bath that keeps the material stable until it cross-links and is stabilized another way, and then you can get the scaffold outside. It's also suitable for making really complex geometry, so with overhangs, for example. And then there's also, it's also possible with extrusion-based techniques to combine it, with microfluidics or with more complex nozzles that will allow you to either make, tubular structures in one step. So, for example, here, if you take a look at the image, you have a coaxial or core shell nozzle. You could also call it, where one material or where two materials are extruded at the same time. One is extruded through the inner nozzle and one through the outer nozzle, and then they cross-link at the interface. And depending on where you extrude, let's say the bioink, if it's in the middle, you stabilize it directly upon deposition. So even if it's liquid inside, it stabilizes at the tip of the nozzle. Or if you would invert it, so you would have the bioink in the outer nozzle, for example, then you could actually print a hollow tube in one step. And yeah, also the implementation of microfluidics, for example, this would allow you to make really complex bioinks where you change the composition during the fabrication process, so you can integrate really chemical gradients into the scaffold itself. So this is, I think, maybe the biggest benefit also or the biggest advantage of extrusion-based techniques. All this versatility in terms of materials and internal composition that you can adjust during the process. The second group of techniques are droplet-based techniques. So what you do here is really apply. This is kind of similar to extrusion-based. You have a nozzle and you press material through it usually, but you do it so fast that the surface tension is broken and you have the material dispersed into individual drops, which are very small. And you can go even into, let's say, into the size of individual cells, for example, which is typically not the case with standard extrusion techniques. So there's different methods of driving the material through nozzles, like inkjet printing or piezoelectric printing. But there's also laser-based techniques that are used to make droplets. So an example of that would be laser-induced forward transfer, where you really you focus pulses of a laser on a wet surface with the bio-ink, and then you locally heat up the surface, which accelerates basically droplets of the bio-ink away from the membrane and towards a target surface. So this is also the droplet-based techniques have been one of the first that were successfully used really for printing living cells. And now they're becoming more sophisticated, but they're kind of more limited in terms of the range of materials that you can use with them. So you cannot use high viscosity materials usually because you have clogging or you don't have really droplet deposition. But it has higher resolution, for example. So there's always a trade-off between what kind of techniques you use. And you have to take that into account when you're planning, let's say, for certain experiments. And the last group of bioprinting techniques that I want to talk about is photolithography. So I could have used the term laser-based techniques, but as you saw on the slide before, laser-based techniques can also be used to making droplets, and lithography is not really what this is. So what happens here is you take a focused laser beam or light source, and in the point where it touches the material, it polymerizes through a chemical reaction, basically. And you're maybe familiar with this. So here on the top left, you see an image of how this works. So you have a stage that is really soaking in resin with photoinitiators, and from the bottom you have a laser beam that is moving and drawing basically the image, and then structuring the scaffold that way. So photolithographic, or we can also say stereolithography or SLA techniques, is also kind of used often for this group of methods. This is also really interesting because it allows really high resolution and structuring of complex scaffolds. But it is kind of limited in terms of materials that can be used for that. So usually the photoinitiators, but even the monomers are within the resin can be toxic, and the degradation products of the scaffolds are also kind of toxic. So there's a trade-off there, but with the development of new materials, this problem has kind of been solved. On the other side, the main advantage of lithographic techniques like this is the really high resolution. So standard SLA techniques like this, it's similar in resolution to, let's say, droplet-based or extrusion techniques. So you are in the range of maybe 50 to 100 microns. But for example, if you exploit multi-photon lithography or two-photon lithography, which would in principle be similar to, multi-photon fluorescence, if you're familiar with that. So you have a reaction only at the focal point of the laser beam, where you have a laser that has longer wavelength, and in the focus it adds up as if you would have you would have a photon with the appropriate wavelength to start the reaction, and you get really fine resolution through that. So for example, here you can go into submicron resolution. So if you look at this image by Kla et al, you even get below 100 nanometers of precision, which is really impressive. And then there's also other lithographic techniques that have started to develop fairly recently. So this is kind of also interesting, the volumetric bioprinting. So there you don't have a focused laser that causes the polymerization, but you have like the polymerization that is the product of temporal superposition. So you have a container with the resin, that is rotated slowly, and then you make a projection of the image with different intensities, and then only where the intensity, you know, accumulates over time appropriately, you have polymerization there. So this is kind of interesting because it's a lot faster in terms of processing when you compare it to SLA like this. So if you have a resolution, let's say of 1 micron, ideally, and you have to do it layer by layer, this also means that every layer will be, you know, 1 micron wide if you want to make large structures. And if it takes, let's say, 30 seconds to polymerize a layer, you can imagine how long it takes to actually make a scaffold like this. And this can be a big disadvantage if you're using live cells, for example. So in terms of the bioprinting techniques, I think what is going to be really interesting for the future is how to, you know, take the advantages of the different approaches and how to bring them together synergistically to make new structures. So exploit, for example, the range of materials that you have with extrusion bioprinting, and getting close to the resolution that you have, for example, with lithography. So this is going to be really interesting to follow in the next years. One of the very important aspects also of scaffold fabrication using any kind of technique really is also the selection of materials. And Murphy and Atala have made a kind of nice set of criteria that are important. They're kind of overlapping, as you can see. So it's really important for materials to have biomimetic properties. So they should ideally mimic the structural and chemical properties of the native ECM to give the cells really the environment that they have in native tissues. They should, if they cannot be the same materials, which often they are not, then they should have, you know, the appropriate biodegradability. So ideally, the material should degrade with the same rate that the cells can kind of replace it. If you have to print it, let's say using extrusion-based techniques, then it has to be printable, so to speak, which means it has to hold its own weight until the process is finished. So if it just, you know, liquefies and isn't stable enough, then you don't have any resolution, and you cannot control the positioning of material and cells. It should have, you know, the chemical effects that cells allow to attach to. It shouldn't have any undesirable effects. And also, yeah, the stability also, or the mechanical properties can also have an impact on how the tissue develops in the long term, and how cells behave on that. So now I would like to shortly go over how we work in this area. So the Bioprinting project started at IRNAS roughly 4 years ago. And in the beginning, this was really an assembly of off-the-shelf components, which we put together, trying to make, you know, scaffolds from some materials and see how it works, how the process works, what are the parameters that you need to control to go from, let's say, liquid material in the cartridge to stable material after deposition, finding all the, you know, the equilibria that are required. And then slowly, in an iterative process, we made it more complex and more refined. And here also, I think, is this is a good example of how the collaboration process worked between the faculty and IRNAS, where at the faculty we had, you know, the practical requirements for making scaffolds and could provide the direct user experience and test what is useful, how the printer can be used in a laboratory setting. And on the other hand, it was nice to have, you know, the technology partner that could really rapidly adapt the system to the needs of the research itself. Yeah, so in the course of 4 years, for example, we came to the device that you see here. It's still not, let's say, a finished product. It's still evolving slowly forward, but we're adding new tools and new capabilities. And for example, one of the projects that we're working on together is development of new extrusion heads and the nozzles. So here, for example, we are trying to put together the material science that we have at the faculty and the requirements for the printing, and mixing them together and developing the appropriate extrusion system that allows us to really control the process, putting together the materials over time in a sequence that is necessary, for example, to make certain type of structures. So here are a few examples of channel-based scaffolds that we made using this nozzle prototype. This is also now including a basic, let's say, microfluidic system, so you can mix up to 3 materials at the same time, and then use it either as a core or a shell material for scaffold fabrication. So this is basically the aspect of 3D bioprinting that we are working on at IRNAS and at the faculty. Now I'd like to go more into other aspects. So let's say after we have made some scaffolds with cells and we want to culture them. So the difficult way to culture cells would be in incubators such as this. So you put them in flasks with, you put samples in flasks with culture media, and you just incubate them at 37 degrees Celsius and 5% CO2. But if you have larger scaffold or tissue samples, this kind of becomes insufficient in terms of nutrient gradients. So you have to implement some kind of active medium exchange. I know that at BioSense you're also working with the Good Food Institute on developing sensors for other reactor types. So you're probably even more familiar with that than I am. But I would still like to give an example of what we've been doing in this area. So I think that was 2 or 3 years back, we started a collaboration with the New Harvest Foundation. So they had an ongoing project really where they wanted to make pork meat using some target scaffolds that they were designing, and they wanted us to build basically an incubation system specifically for that. So we had a target scaffold, and they provided us with the requirements for the type of perfusion and the type of control they would need for that perfusion. So we made basically a custom culturing container and a perfusion system, and even an incubator at that point that would house basically the whole thing. So the incubator looked first like this. So this is a recycled wine cooler really that has temperature and CO2 regulation, and inside you have perfusion trays which are really syringe pumps that you can control remotely and adjust, let's say, perfusion speed. And there's also room for sensors, for example, to see what the dissolved oxygen content is inside of the medium, for example, and the input and output. But as we worked on the project through time, we kind of noticed that, for example, the incubator part wasn't really necessary, because all the labs that work on tissue engineering or cell culture in general, they already have incubators. So it's much more feasible to really work on the perfusion type and improve the control and then the sensorics that's kind of connected to that. So at the moment we have a perfusion system that is under let's say validation at the faculty. So we have a tray, we have an incubator microscope where we can see what's happening with the cells over time. And we are now actually expanding the project even more. So now we are starting a broader collaboration between New Harvest, the faculty, and IRNAS, where we are designing a really, let's say, more broad experimental setup with the idea to test and see the synergies of different gradients within tissue samples. So what we want to do is design scaffolds that are 3D printed, that have implemented chemical and structural gradients. So in terms of stiffness, for example, we want to control perfusion and the chemical gradients that goes, let's say, in another direction, and also implement other gradients and see what happens, you know, in the micro scale at the intersection of different gradients, see if there's any synergistic or antagonistic effects between them, and how cells behave, how the phenotypes form, depending on, you know, their spatial position within scaffolds and stuff like that. So we are really starting with this at the moment. So this is going to be more interesting to look at maybe in 1 or 2 years from now. But yeah, I hope to be able to present more in some time. And I think this is the most of the stuff that I wanted to talk about today, but I would be happy to answer any questions that you may have. So thank you for listening, and yeah.
Thank you very much for a nice presentation.